So to start with the first experiment, it is to study the osmosis by potato osmoscope. Now here the requirements which we need is what? One potato, then pillar or the scalpel. This is a scalpel. Then the saffronin which we will be using to use the solution with water, and then this is the sucrose solution. Now first we have to just take a fresh potato tuber and peel off the skin with the help of what? The scalpel or what? The pillar. Now I'm just removing the skin. Don't think that I'm preparing the vegetable. Okay. So otherwise you all will think that I'm preparing the vegetable. It is the potato osmoscope which you have to do the experiment. Now I have removed all the skin. I'm keeping it aside. Now you have to give a cut. Now from between you have to just scoop it. Cut the potato from one side in such a way that it will make a flat base. Means a flat base should be there. You have to keep in mind that the down portion, this portion, there should be no hole. Only upper part it should be cut properly. See here, we have scooped with the pillar, the tuber properly so as to make a hollow block in the potato with a thin intact base bottom, and a care should be taken that it should not rupture at the base part. See, now what we will do, this we will keep it aside. Now this is the petri dish. We have to take the water. In this water I am just adding little bit saffronin. This is what the saffronin. We have to make the water colored solution. 1 drop is enough. Okay. Now what we will do, now add concentrated sugar. Now this is what the concentrated sugar solution in that cavity and mark it level. Now what we have to do, just take the potato and then you have to just add this solution and mark the level and keep it inside this Petri dish. Now you can see the level. Now you have to place this potato osmosis in the Petri dish and fill with what the colored water and keep it for half an hour. Afterwards you can see the rise in that potato cavity part. So now you have seen the level in the hollow cavity. Slowly and slowly it has been risen. So the level in the hollow cavity, can you see the pin? Yes, and the solution also, it has been become as what the pink in color. Now I will mark the level here. See the level, the new level. Okay, so it is the observation which you have seen means what? The level of the sugar solution in the potato osmoscope rises after some time, and the solution in the cavity osmoscope also becomes what? Colored. So that's why you have seen this is what the colored solution in the hollow cavity. And it can be inferred that the colored water from the Petri dish, which I am using the Petri dish or the bowl, has entered the cavity of the potato osmoscope. So the conclusion I will say here means what? The rise in the level of the sugar solution. So here which solution we have taken? The sugar solution is because of end osmosis. Now end osmosis, as I told you, it can be seen in hypotonic solution, and the hypotonic solution means what? The higher solute concentration which is what? Inside than the outside. So the molecules of the water moves from what? The region of the high concentration toward the region of the low potential through what? The differentially permeable membrane of potato cells. So this was all about what? The experiment of osmosis using potato osmoscope. So to start with practical 1, experiment number 1, study of osmosis by potato osmoscope. Now you all have seen the video how the experiment is performed. Now first of all, the aim to study the process of osmosis using potato osmoscope. Now the requirement, as I told you, the fresh potato is used, then the pillar or the scalpel, then the Petri dish you can take, or a bowl or a shallow glass which should be transparent, then the beaker you can take, then the pins and concentrated sugar solution or sucrose, and the color water. So in color water you can add saffron in. Now the principle, what is the principle behind this experiment? So when two solutions of different concentrations are separated by semi-permeable membrane. Now what is a semi-permeable membrane? Means it is a biological membrane which functions by permitting the movement of certain molecules or ions to pass through it. Means the ions will be passing through it. So when two solutions, if you are taking of different concentration, so one is high or low and the other low, are separated by semi-permeable membrane, the flow of the solvent in the is from the region of the weaker solution, that means having low solute concentration, to the region of the stronger solution having the high solute concentration, till the equilibrium is reached, means till it is balanced, so that the osmotic pressure are balanced. So they are of two types, as you have studied long back, end osmosis and ex osmosis. Now end osmosis means inside and ex osmosis means what? It will be outside. Now end osmosis means what? When a substance is placed in a hypotonic solution, Now when I am talking about the hypotonic solution, now this is a diagram where you have seen the beaker. Here they have taken beaker, I have used the petri dish, and in that the potato is being taken, which is down flat and then in between hollow, and in the hollows we have taken what the concentrated sugar solution. Okay, and then after that what we have done, we have, with the help of the pin, we have marked it, and this is the peeled potato. Okay, then later on what you can see, there will be a rise in the water level in the hollow part. Okay, so this diagram is important, which you have to draw in your book on your left hand side in your journal. Now the observation which you see after 15 or 20 minutes or 25 minutes, it will be seen that the level of the sugar solution, see the sugar solution, first year mark, the marking was there, now it has been raised. So the level of the sugar solution in the potato osmoscope rises after some time. The solution in the cavity osmoscope also becomes what? Colored. Means this part which is there, it becomes what? Colored. And it can be inferred that the colored water from the petri dish bowl has entered the cavity of the potato osmoscope. Now as I was telling you, and you all have studied also, osmotic solution, there are three types, that is isotonic, hypertonic and hypotonic. Now isotonic means what? When this concentration, okay, it will be same concentration of the solutes will be present inside as well as outside, because the inference you will be having a look here that it is what? End osmosis. So that's why it should be clear what is end osmosis. Now hypertonic means what? Where one of the solution, the solution is one that a higher solute concentration outside than the cell inside, and hypo is what? This is the solution where it is having the higher solute concentration inside the cell. So here it is what? The inside the cell there is what? The higher solute concentration from the outside. So it means what? It is hypotonic. It is what? Hypotonic. So end osmosis, now you can see the conclusion here. The rise in the level of the sugar solution is because of end osmosis. Now end osmosis means what? When a substance is placed in a hypotonic solution. Hypotonic solution means it is one that a higher solution, it is one that having the higher solute concentration inside the cell than the outside. Means inside it will be what? The higher solute rather than what? The outside. And whereas the X osmosis, it will be totally different. It will become here a flaccid type. So water molecules moves from the region of the higher water potential in the petri dish to a region of the low water potential sugar solution in the potato cavity. So in the potato cavity, through differentially permeable membrane of the potato cells. So this is what about the potato osmoscope experiment. And then next which you are going to study is about the significance of it. The significance here, if you see, is what? Essential for the survival of the cell, and it is involved the movement of the water molecules between the cell and the cell organelles. Okay, so thank you.